Hello, this is Mr. Unger, your biology teacher, and this video is to guide you through assignment number 8, which will be a lab. Assignment 8, Unit 6, Session 9, Lab 2, Photosynthesis, specifically light wavelength and photosynthesis. You're going to be investigating how changing the wavelength of the light that the plant is exposed to modifies how much growth you observe. Essentially, what you're testing for is what light wavelengths promote the most growth and what light wavelengths least promote growth. So first thing, when you click on that, you'll follow the link to the Google Doc. I have that open already. It'll take you here, and the first thing I'd like you to do when you open up this Google Doc is to, I use a template to reuse the same thing over and over again, change student name to your name. And then on the second page, you'll follow this link. I'd suggest that when you follow that link, it will take you to this page here, the page where we have the virtual lab. You might want to consider, at least for parts of the lab, if not the entire time you're doing it, to split your screen in a sense. Here's what I mean. I'm going to minimize the size of this window. And I'm going to also take, let's see, I'm going to take the document that I'm working on, and I'm going to put that side by side so that I can simultaneously view the lab, or at least some of the information from the lab, and I could put my cursor on the document and perhaps type something up. That's really helpful if you're trying to modify some text that you saw and make it your own so you avoid plagiarizing it. That way you can just look at the text, think about it for a moment, type your own version of it, and keep on going. So that's a good strategy to apply not just in this class, but other classes as well. That may be something that you do all the time already, but it's a good way so that you can look and take the text information and translate it to another document. All right, let me go back to the view I had previously. All right, so you'll notice that the objective section and the introduction section are missing text. Your job is going to be to view this virtual document and start by reading the title, which colors of the light spectrum are the most important for plant growth, and then reading all this text. It's going to give you the background information for this lab. It's not going to go ahead and answer the question and give away which colors will be the most productive for the plants, but it's setting you up and giving you the principles behind this. Please read that and give a summary of those 3 paragraphs there, essentially, for your introduction. And the objective, I oftentimes wait until very late in my lab write-up process before doing an objective. Writing the objective might be something that you do almost the very last thing, the same sort of time when you're doing your discussion, your conclusion. The objective is essentially a condensed version of the conclusion, where you're going to state just very simply what your goals were. Like for this lab, the objective is going to be something like to investigate which wavelengths of light most promote plant growth. The key question is really like, since plants are green, does that mean they absorb green light or they reflect green light? Would green light be effective for them for photosynthesis, or would green light be ineffective? You're kind of thinking on those lines. I don't want to give away anything. You'll make your own hypothesis, and the hypothesis, it's set up with a question right here that you essentially just need to answer to give your hypothesis. Here are the materials. You'll want to list those in bullet points. You can just look here on what was being used. It may not give an actual materials list here. It does give a procedure, but it doesn't give a materials list. It'll be up to you to observe what was being used here and to write those things down as your materials. I'm seeing spinach plants, radish plants. Well, actually, they were seeds that were planted. Spinach seeds, radish seeds, lettuce seeds. I'm seeing these plant pots. I'm seeing a measuring stick. I'm seeing grow lights. I'm seeing a grow house overall. I'm seeing the ability to use various colors of light, so we have various color light bulbs that we'll be using, and a timer. I guess that's about it. So that would be a pretty full list of the materials here. And then the procedure. This is the one place where I would not mind if you copy and paste. Well, I guess you're not going to be able to copy and paste, but you could split it up side by side like I was showing you earlier, and that'd be a good way to record the procedure. The only procedure is what I'll allow you to copy word for word. Anything else, it's considered plagiarism. Please don't copy and paste or write text out word for word identical to what you read from a source. Always modify it. This is the one exception. In the procedure, I will allow you to duplicate the procedure in that virtual lab word for word, but that's the only section where you can do that. So here's how you're going to go through that procedure. All right, let me reset this. So each round, I'd say start with, it doesn't really matter which plant you start with. Okay, so I clicked on spinach. It planted spinach plants in all 6 of those. And since I have 2 different grow houses, I want to make the conditions different in those 2 grow houses. So one thing I can make different is I can use different colors of light. One of them is red, the other one will be grown with orange light. I'll turn the lights on and the experiment begins. It goes fast forwarding through how many days? 30 days. And these are the results after 30 days of growth. The way you're going to quantify these results and record your data is by using this ruler. Please, the ruler is in centimeters. Please record to the nearest 1/10 of a centimeter. You can drag this ruler around, put it right next to a plant to give an accurate measurement. I'd say this first one measures 15.2. No, maybe more like 15.3, somewhere around there. 15.3 centimeters. This next one, 21.5 centimeters. The third one, I'd say that comes out to 17.5 centimeters. You're going to want to average those 3 values, and you'll give one averaged height for this particular plant, the spinach plant, grown under red light, and then an average height for when spinach was grown under orange light. Once you've taken your measurements and recorded your values, you're going to be recording the values in 2 places. One place you're going to record them is on the lab document. You'll put them in this data table, but it's also going to be important that you put your data in the data table that's in the virtual lab. You'll go down to the lower left-hand corner and click on Table, and you can enter your data in here. Let me just make up a number. Let's say the average was 12 for red light, and let's see, what plant was that? Okay, so the spinach. Let's say that grown with red light, it averaged 12 centimeters. I've entered my data point there. It's the same data point I would have entered in the data table on the virtual lab document. Once you've completed collecting all the data, that means for each color, you'll test out each color for each of the 3 plants. And once you've recorded all of that data, you'll have it all recorded on your document in that data table, but also here. And the reason you want it here is so that you can click on Graph and create this graph. Since I only entered 1 data point, that's all that you see here, but yours should be complete, and it'll show the patterns that you've now been able to observe, that you've recorded. You'll probably want to take a screenshot of this. You could click on Print, but the thing is that's going to save like a full page of it, and that takes up more space in your lab document. It's not a big deal, but I'd say the better option is, instead of clicking on Print, find a feature on your computer that allows you to take a screenshot. For me, it's something I have in my toolbar at the bottom of my screen. It's called Snip and Sketch on my computer. So I'd click New, take a new snip, and then copy that and paste it into the document. And the location where you'll place that in the document, let's see, let me close this out. Back in the document, you'll want to paste that here where you see the yellow highlighted text. I gave that as instructions. If ever you see a bracketed text that has the highlighting on it, I'm trying to make it stand out so you cannot miss it with your eyes. It's giving you the instructions, but please, once you've pasted your data table there, or excuse me, once you've pasted your graph there, delete that highlighted text. That's only there to catch your eye, so you'll follow those instructions. It should not be left there in your final lab report. Okay, so once you've done all this, you've collected the data for each of the plants under each of the light colors. You've generated your graph by putting the data not just in this table, but in the table in the virtual lab. and you've created the graph in the virtual lab and transferred it here, the last thing to do is to answer these journal questions and then write your conclusion. Now, I don't think the journal questions need any explanation. Of course, the rule is no plagiarism. Don't just copy and paste answers in there, because I know you'll likely be able to find exact answers to this if you Googled it. That's the reality we face. Do not do that. Do not plagiarize, please. It is illegal, and it would result in a failing grade on this. The conclusion. What I'd like you to do for the conclusion is summarize overall what you observed in this lab, and make sure that you include a quote from the textbook. It does not have to be from a specific chapter, but find a quote that supports what you found from this lab. And I want you to use that quote and give credit to the textbook as your source for that quote. You don't have to use quotation marks around that quote. Type out that text and follow it with a parentheses that lists the title of, or I'm sorry, the last name of the first author listed, followed by comma, the date that the textbook was published. You can find that by going into the textbook and looking at like the first few pages of the textbook. It'll show you who the author is and what year it was published. So please find that information, and whatever passage you choose to quote from the textbook, follow it up with the shorthand citation in the text of the conclusion, where again you follow the quotes without quotation marks. You just follow, at the end of the sentence you have a parentheses, and you have the leading author, the last name of the leading author, comma, the year that the textbook was published. And then in the references section is where you're going to give the longer version of the citation. You're going to give a bibliographic entry there, and there are plenty of websites that will assist you in making bibliographic entries. There's easybib.com or .net, I'm not sure which one it is, but there are plenty of those websites. EasyBib is one of them, but there are others as well. All of them are easy to use and can generate the citation for you. So you take advantage of that, use one of those websites that will generate the citation for you, and you'll give the shorthand version in the conclusion, and then the extended version of it that you got from entering the info about the textbook onto the website, whichever one you choose, like easybib.com. That information can be copied and pasted and inserted in the references section to give credit to your textbook, having cited some text from that textbook. That does it. I think that's the last bit of information that you needed for this. If you have any questions, feel free to contact me, and I wish you luck with this lab.